thanks everyone for joining us today. This is the last PHIND symposium of the year, so really great to have everybody with us, and I hope some of you were able to join the PHIND symposium last week. So for today, I'm very excited to hear about Allison Zhang's PHIND work that she's been working on as part of Mike Snyder's lab. And so I'll give a brief bio on her. Dr. Zhang joined Michael Snyder's group in 2018 at Stanford University after receiving her PhD in pharmacology and toxicology from the University of Kansas. Her research focuses on understanding the human exposome, particularly how the exposome affects human health. She is interested in applying multi-omics approaches using high-throughput sequencing, mass spectrometry, and wearable devices to understand the exposome and its health consequences at the personal level. She has studied the human exposome in multiple environmental settings, including hospital and during wildfire, and in complex disease settings such as Crohn's disease and autism. Her current work also includes developing wearable sensors to profile personal exposome that aid precision environmental health. And her talk will be on a powerful personal exposome tracker for precision environmental health. So I'll turn it over to you, Allison. I'm really looking forward to it.
Thank you for the next introduction, Ryan. It's a great pleasure to be here. And today I'm going to introduce you my latest work. Let me share my screen first. Okay, there we go. Everyone can see my slides?
Yes, looks good.
All right, great. Yeah, so today I'm going to introduce you my work on the powerful exposometer for precision environmental health. So the first one I want to introduce is what is the exposome. The exposome concept was first defined by Christopher Wilde in 2005. So by definition, it encompasses life course environmental exposures, including lifestyle factors from the prenatal period onwards. So it covers a large amount of the stuff around us, and it includes many aspects of our life, such as the ecosystem that includes the green space, the population density, lifestyle that includes diet and exercise, social network, social income, and also the physical chemical domain, which most of us are the most familiar with. That includes the air, water, soil, and all the personal care products we use, pesticides, and all that stuff. So there are two types of the exposome. One is the biological, and the other one is the chemical. The biological exposome includes fungi, bacteria, plants, and pets. And the chemicals includes all the paints, pesticides, carcinogens, and personal care products. So why do we study the exposome? The short answer is it impacts health. So there are many components of the external environment, and they will interact with each other, also interact with our internal environment to impact our health. A lot of the diseases have known external triggers, such as lung cancer and cigarette smoking, pollen, and allergy. However, there are still a lot of diseases that has no exact cause. Increasing evidence suggests that environmental factors may play a role in the disease etiology. So studying the exposome will help us better understand how those diseases happens and may provide a new avenue for disease management and even prevention. So our lab is particularly interested in the airborne exposome. And the most well-studied airborne element is probably particulate matter, also also known as the PM. So they are the small droplets in the air that contains liquid particles and solid particles. You may have heard of the most notorious PM 2.5. That 2.5 refers to the diameter of those droplets. So 2.5 refers to anything smaller than 2.5 microns. And PM 10 refers to anything that's smaller than 10 microns. So once inhaled, the PM can deposit into different parts of the respiratory tract. The PM10 can stay in the trachea, and PM2.5 can go deeper into the lung, into the bronchioles. The even smaller PM1 can go through the olfactory nerve and go into the brain to affect brain dysfunctions. Short-term exposures to PMs are associated with coughing, sneezing, runny nose. A long-term exposure is linked with impaired function in the lung and the cardiovascular system, and even death. Traditionally, we study the particulate matter using those monitor stations. Those are shed-like structures outdoors. There are many drawbacks associated with these monitor stations. Number one being a low resolution. So there's usually only one in a large geographic area. So you cannot provide a precise personal exposure information. For example, if I fire up my barbecue right now, a monitoring station 20 miles from me is not gonna report this exposure. And the second drawback is low information. The monitor station will only report a total concentration of those particles and some gases, but it has no information on exactly what particles are present, whether there are carcinogens, pesticides, or allergens and pathogens. So this could be health relevant to many individuals. So following this scheme, we propose the personal exposure monitoring. So we use a wearable device to profile the comprehensive biological and chemical exposure at the personal level. So there are two components that makes the wearable unique and able to capture both the biological and the chemical exposure. We re-engineered a wearable device that were previously used only for PM2.5 studies to include two unique components. One is a PES filter that we use to capture the biotics from there. That includes pathogens and allergens. We then extract DNA and RNA from those filters and then use next generation sequencing to analyze the biological exposure. The other part is a 3D printed housing for zeolite absorbance that we use to capture the chemicals from the air. That includes carcinogens and pesticides. We use mass spectrometry to analyze the chemical part. So using this paradigm, we followed 15 individuals for up to 890 days. The participants were asked to wear this device on their arms or place on the table near them to capture their personal exposure. Each sampling period lasts about 3 to 5 days. Here we can see that P1, P2, and P3 were heavily monitored, and their samples covers all 4 seasons. So our own analysis mainly focused on the P1, P2, and P3. Our participants were also asked to use the MOVES app to track their locations during the study so that we can see if there are any influence of the location on the exposure. So here shows their locations during the study. P1 is shown by the green dot, which we can see travels all over the world. P2 mainly travels around the Bay, and P3 is mainly travel through the South Bay and San Francisco. So what did we capture? So with this paradigm, we are able to capture more than 70 billion reads and more than 7 million assembled contexts. And with those contexts, we identify more than 25,000 species that covers all domains of life, including bacteria, fungi, animals, plants, and viruses. We also did some of our findings using electron microscopy. On the right shows some EM figures of the pollens and the spores that were trapped by the filter. We first analyzed the composition of the exposome to see what exactly is there. So we took our samples and classified based on their kingdom, or subkingdom. So the top row shows the exposome composition by timeline, and the bottom one shows the composition by clustering. So if we can take look at the P1's samples, we could see those samples, the compositions are different across the study. Some are mainly of the amber color, that's the fungi, and some are more of the green colors, that's the plants. So if we cluster those samples by the dominant kingdom of each sample, we could see some of the samples were mainly plant dominated, showing as this green group, and some are bacteria dominated, some are fungi dominated, and some are mixed. The P2 and P3 also have a similar trend. So we could see the composition of the exposome is highly dynamic and is very diverse. We next focus on what's the source of variation on the exposome. So we took P1 samples and did a PCA analysis on those samples to see whether locations have an effect on this exposome composition. So the top one shows the P1 samples against location. Those are broadly defined areas such as Asia, Europe, and Oceania. We found that samples from the same location are clustered together. And it's similar with seasons. Samples from the same seasons tend to cluster together. So this shows that location and season may have influence on the exposome composition. So we identified there may be 3 variables that could cause this. Number 1 is the environment, that includes season, temperature, humidity. And number 2 is the spatial lifestyle factors, such as location. And number 3 is the technical artifacts. So we use the partial distance-based redundancy analysis and assign all of our meta variables into these 3 categories. When we found that spatial lifestyle and environmental factors explains most of the variations. So this shows that location and season have an influence on the diverse exposome. So to better understand how the location influenced the exposome composition, we did a sub-experiment where we monitored 4 participants for 1 month. All of them live in the broad Bay Area, but they have their own unique work-life routines. P1, as always, travels all over the world. P3, shown as the blue dot, travels across the Bay. P5 is the green dot, mainly carry out his activity in the South Bay, and P6 is mainly reside in the San Francisco. We could see that samples from each participant tend to cluster together, and P1 samples is spread all over the place because he travels a lot. This tells us that even for people living in the general Bay Area, their exposome components is still different. And for those who are interested, we found some sludge-associated bacteria in P6 samples, that's San Francisco, and P1 was mainly exposed to plants and fungi. We also investigate deeper into the effects of seasons on the exposome. So undoubtedly, the seasons will play a huge role in the environment, different plants, flowers in different times, and this will definitely affect the exposome composition. So we use a fuzzy Siemens clustering to identify organisms with seasonal patterns. We found 4 clusters, and each display a peak in specific season. We found the winter, shown as the navy color, is the largest cluster with 121 genera. On the right shows a couple of examples that has a unique seasonal peak. On the top shows pine trees. The pine usually sheds pollen in late spring and early summer, and we detected a peak at this corresponding months. On the other hand, the fungi, false turkey tail, spore seasons around fall, so we see a peak during that months as well. So this shows that biological exposome is affected by location and seasons. So what about the chemicals? With our pipeline, we detected over 3,000 features from our samples, and the top categories being pesticides, personal care products, plasticizers, and carcinogens. We next look at whether those chemicals are influenced by seasons as well. So we have two examples here. Both of them are from plants. So these, the top one, we could see its concentrations peak during late spring, shown as the red bars here. And the Allotine, this is the bottom chemical, shows a peak during winter time. This shows that some chemicals have seasonal patterns as well. But we also found some ubiquitous chemicals that are present in all of our samples. Those include plastics, DET, pesticides, and carcinogens. And next, we look at the dynamics between those chemicals to see if there are any interesting correlations among them. We found this one that's called geosmin. That's the earthy smell when raining. And omethylate is a pesticide. These two chemicals were highly positively correlated. And those samples were collected during the rainy periods. So this suggests that these compounds can accumulate on the ground and then release into the air raining. Phthalate, on the other hand, is a synthetic plastic component. It can deposit into the air by absorbing onto the suspended particles. So it is expected to decrease during rainy periods. And we also found this phthalate is negatively correlated with the above two compounds. We then examined the relationship between the biological and chemical exposures. Are there any correlations among them? We found an interesting pattern that's linked by this chemical pyridine. It's a very common organic solvent. It's widely used in paint and dye. And we found this is in an it's anti-correlated with a number of fungal species. So we found a very low pyridine exposures in the campus home positions of this P1. P1's house was painted using low VOC paint, so it's very greenhouse, but P1 was exposed to fungi. And some of those species are human pathogens. But these fungi were found at lower concentration where pyridine is high, such as in the Boston area. So just be careful when you consider if you want to use pyridine as an antifungal agent and add it to your paint, because pyridine itself can cause a lot of health problems. Short-term symptoms such as nausea and headaches, long-term symptoms such as liver and kidney damage. So just some interesting stuff to consider when you paint your house. So far we've demonstrated that exposome is highly dynamic and diverse. Season and location has a huge impact on the composition of the exposome. And also there are complex interactions between the exposome components. So with the wearable device that we use in the study that we re-engineered to fit our purpose, there are many disadvantages of those devices. Number one being super expensive. So the Gen 1 showing on the left is a microPMs from RTI. So that one is $2,500 per piece. So it's very expensive, and it's really hurts a lot when some of your participants lose one of those devices. And another thing about this device is have a very low flow rate. It's only 0.5 liter per minute. We want to care about the flow rate because we want to have a flow rate closer to a human breathing rate, so that when it comes to quantification, we have a better evidence of this exposure. So the After the micro PEM, we found this UPass shown on the right. This bike device is only half of the price of the micro PEM, but still at 1,300 per piece. But unlike the micro PM, this UPass does not have any real-time measurements of particles. So that it means it has no real-time measurement of PM 2.5 or PM 10. Although this is not the most comprehensive understanding of the airborne exposome, the PM concentrations is a good indicator of air quality and is widely adopted by the general public so that they know they have to act when the PM concentration reaches 200 and a smaller PM concentration means a cleaner environment. The flow rate of the UPass is increased to 2 liters per minute, but still lower than average human breathing rate, which is about 8 liters per minute. Sorry, 11 liters per minute. So the goal of my project is to develop an exposometer that is low cost and has a flow rate closer to human breathing rate and has multiple functions. Number one, giving out real-time particle measurements so the users could act upon these real-time information. Also have the components to allow for comprehensive exposome analysis. The device has to be portable, wearable, and also has to have an app to provide real-time data and also to build a foundation for the exposure map, which I'll explain later. So here's the design overview of the device. So it's a very straightforward device. It's not rocket science. So the air will come in and that go through the dust sensor. The dust sensor will provide a real-time measurement of PM concentrations. And then at the end of the airflow, we have 2 filters. One is the PS filter and one is the zeolite absorbance. The PS filter will be used for collecting biological agents. And then we use next generation sequencing to analyze the biological exposome. The zeolite will be used to capture chemicals from the air, which will then be analyzed using mass spectrometry for chemical exposome analysis. And the information will pass on to the app to display all the measurements from the device. And also there will be other sensors on board, such as GPS, to help understand how the exposures across different locations, and also temperature, humidity, and gas sensors on board as well. I was trained as a pharmacologist, so starting this project as an engineering project is really challenging for me. I had no idea where to start. So I turned to Google to see how I can build such a device. So luckily, with the development of IoT, right now I found that using Arduino boards and the sensor modules, I can get a prototype that this just shows proof of principle ideas of whether this one will work. So on the left shows the prototype that I have at the time of assembly. So with I have an Arduino board with all the sensors across the picture. So on the right shows a measurement from this device. It shows the temperature, humidity, and PM concentrations in real time. So this demonstrates, well, all of these can work. So what's the next step? So I talked to the Stanford prototyping facility. I got Allison on board to help me condense everything I have on the lab fixture into a printed circuit board and fit it into a small casing so that it can be a real wearable device that participants can take this device with them to capture their personal exposome. So the developed device, which I call it a PAC, that stands for Personal Exposome Tracker, is this a great picture we have on the left. And it's a very small size device. So if you have an iPhone, think of if you fold it in half, that's how big the device is. The device is super lightweight, it's only 100 grams. It measures all the And traditional parameters of an air quality sensor, such as the particulate matter concentrations, temperature, humidity, VOC, GPS, and air quality index. It also allows the capturing of the biological and chemical exposome so that we could analyze the most comprehensive personal exposome. Some of the specifications and of this device here shows the particulate matter captured by this device. The device was left on the porch of 3165 Porter Drive, and then we let the device run for 40 minutes. Every half a minute, the device will report a PM concentration. We use 5 different devices for this experiment. You can see the device could provide real-time measurement of PM1, PM4, PM10, and PM2.5 concentrations, and also the variability between different devices are quite small. We also test the device ability to capture the exposome. So with our addition of the 2 filters inside the device, we left this device outside on the porch again for 5 days to capture the exposome. After we sequenced all of our samples, we found this device is capable of capturing the exposome. Here shows the biologicals captured by the device. All of the domains can be captured by this device, and our sample shows at the time of collection, bacteria is the most dominant kingdom of the biological exposome. On the chemical side, we use our samples to go through the mass spectrometry and correlate again our database. We find thousands of chemical features captured by this device. Because this is our untargeted analysis, so our confidence level is not as great as we want it to be. For the validation, we'll need to get a set of standards to further validate what exactly we captured. Here is the interface of the Exposometer app. So right now, the app is mainly used for displaying the data that collected by the device. It will show in real time what is captured by the device, mainly temperature, humidity, and PM concentrations. We are also working on the exposure map view, which will show the exposures captured by the device location by location, so that the users of this map will get an idea of the exposures around a certain location, and to avoid some of the harmful locations for particular individuals. So comparing with other devices, our device is quite small but super powerful. It has a flow rate of 8 liters per minute, huge, a lot better than the previous generations that we re-engineered. Besides the PM2.5 and PM10, the device also have compartments to capture the biological and chemical exposure. Additionally, this device has a gas sensor to allow for detection of VOCs and air quality index. The device also comes with an app to display the data and show an exposure map later in this study. So right now we view the device as capable of collecting the biological and the chemical exposure. But how can we use this data for? We do not want to just identify that a person A is exposed to fungi and person B is exposed to a bacteria. We want to use this information to get better understanding of a person's health. So that comes to the concept of precision environmental health. So the precision environmental health is similar to precision health, which we consider a person's genomic information in addition as a source to provide healthcare. For the precision environmental health, we add in the environmental factors to get a better picture of how this person will react to a certain stressor or certain drug that for a disease. So to demonstrate the capability of using an exposome data to help with precision environmental health, we use a multi-omic approach. So we monitored a participant, that's the P1 from the one that I just, from the previous study I just showed. We use the exposometer to track the exposure components of this participant. Additionally, we collect blood samples from this participant to run for metabolomics, proteomics, blood tests, and cytokines for immune markers. We also collect stool samples for the gut microbiome profiles. So the aim of the study is to see how the external environment affects the internal omes and health. So we first did a Spearman correlation analysis of our samples between the external exposome and internal omes. We found almost 9,000 correlations among them. And specifically, if you take a look at the right side picture, the orange one is the chemical, orange bar is the chemical exposome, and the purple bar is the biological exposome. The biological exposome has the most number of correlations with the internal omes, and the top one being the metabolome, followed by showing green, followed by the proteome showing red, and then gut microbiome showing brown. For the chemical exposome, it also has the most number of correlations with metabolome showing green, followed by gut microbiome showing brown, and then proteome showing red. We then take a look at each of the omes in more detail. So this is our analysis of the exposome versus the gut microbiome. On the left shows the correlations between the exposome and gut microbiome. We found 1,300 correlations among them. The outer ring shows the exposome components, and the purple means biological exposome component, and the orange means a chemical component. The inner ring shows components from the gut microbiome. We found 6 bacteria that has the most number of correlations with the external environment. Interestingly, we found that we found an interesting pattern of these bacteria. On the right shows the genus Allistipes. This genus is involved in inflammation and various diseases. We found this genus is only positively correlated with the chemical exposome, shown with the right line, and negatively correlated with the biological exposome, shown with this blue line. On the other hand, Roseburia, which has been shown to have the function of regulate the gut inflammatory process and amelioration of atherosclerosis, is only negatively correlated with the chemicals, but positively correlated with the biologicals. This suggests that the genus Allistipes may be mainly involved in the pro-inflammatory response, while Roseburia are mainly involved in the anti-inflammatory response when the body is encountered with an external agent. So we next look at the exposome versus the proteome. So again, on the right, on the left, the outer ring is the exposome components. Purple means biological, and orange means chemical. And the inner rings are different proteins we capture. And the lines between the inner and outer rings indicates a correlation. A blue line means negatively correlated, and the red line means positively correlated. We found over 2,000 correlations between the exposome and proteome, and most of these correlations are positively correlated. We then put these proteins into pathways to see what biological processes were mainly affected by the exposome. We found that immune pathways are the most common pathway that are correlating with the chemical and the biological exposure. And similar with the proteome, we also did a correlation analysis with the metabolome. The metabolome, I have no doubt, is the oome that interact most with the exposome, since as the xenobiotics will interact with endogenous metabolites first when it's entered in the body. So we found 4,624 correlations between the exposome and metabolome. We also put the metabolites into pathways to see what pathways are affected by the exposome. We found some interesting patterns with the correlation. For the chemical exposome, for example, we found some of the chemicals show with this blue bar only positively correlated with these pathways, while other chemicals show with this maroon bar only negatively correlated with pathways. But on the biological side, most of these correlations are mixed. There are many pathways that are shared by both the chemical and biological exposome pathways, and they were mainly from the protein metabolism pathways. So we found so many correlations between the internal exposome and internal omics with the exposome, but what does this mean for health? So we next investigate the effects of exposome using routine blood tests. So unlike the internal multi-omics, which has no standard on what is healthy and what is not, this clinical blood tests have established standards that we can use as an indicator of a person's health. We first use an impact contribution analysis of the external exposome on the clinical blood tests that we have. We found that the variation of these blood tests can be 76% of this variation of these blood tests can be explained by exposome components. And more importantly, the top 13 blood tests, which can be almost like entirely contributed by the exposome components, are mainly related to the immune system, liver, and kidney functions, which again that shows that these systems are involved in the body's response to xenobiotics. Here shows some examples of those blood tests. All of these tests were tested 5 times during our study, and all 3 of them have abnormal values compared to the normal range. Take glucose, for example. The normal range is shown by this green belt. We found that glucose levels is positively correlated. The positive correlation is shown by a red box on the left column, and the negative correlation is shown with this blue bar. So the glucose levels is positively correlated with tetrachloroethylene and Acetobacter and other biological agents, but it's negatively correlated with salicylic acid and dibromethine. So the salicylic acid has been shown to reduce blood glucose levels. So this makes sense and validate our results, but most of those environmental components do not have a solid evidence of their relationship with glucose levels. So this requires further validation of the results to see what exactly this correlation means, whether it's a causal relationship or it's just a correlation. Similar with eosinophils and urine nitrogen, they all have certain exposome components that correlate with the levels of these two biomarkers. And this shows that these components could be involved in the metabolism of the exposome from them. So this is very exciting for us. This shows that using a wearable device that we could collect a comprehensive personal exposome profile, and using combine this exposome profile with a person's health indicator, we could see that using a wearable device to profile a person's external environment could provide useful insight onto a person's health. And this will help the precision environmental health. So some future directions for the study, number one is to set up a cloud database to store all of the data we collected from the wearable device. And number two is set up the exposure map, as I mentioned, so that people can avoid high-risk areas. Although the device that we developed is very small but very powerful, but the main drawback of this device is it cannot output real-time exposure information. So it cannot tell you that right now you are exposed to bacteria A, and then 10 minutes later it will tell you you're now exposed to fungi B. So our goal for the future is to develop a device that provide real-time exposure measurements. Of course, measuring everything from the air and get real-time information is impractical. We'll select a subset of the most concerned exposures and provide a real-time measurement of these chemicals. And we'll also get a flow rate that's closer to human breathing rate, that's about 11 liters per minute. And ideally, we will also have measurements from the blood so that we could further relate the external environment with a person's health. Before I end, I'd like to do a promotion of the study that we're doing right now. We're using a wearable device to capture the human exposome in Crohn's disease participants to see how the external environment could affect the disease course of Crohn's, such as flare-ups. There's no cost to participate, and if you know someone who has Crohn's and would like to know about their environment, or if you just want to know about your environment in general and be a healthy volunteer, feel free to contact us. We'd love to have you on board. Also, we are rolling out another study that captures the home exposures versus the workplace exposures to see how the occupational exposures could affect people's health. And lastly, I want to thank lab members from the Stanford Lab who has helped me on the project, Chao and Peng, Xiao Tao and Xiangping and Mark. And also I'd like to thank Allison from Stanford prototyping facility to help me with the PCB work. Also, I'd like to thank the funding sources, the Stanford PHIND seed grant, and also fundings from NIH and SCGPM. I would like to thank everyone for listening, and now can take any questions that you may have.
Great, thanks, Allison. Very interesting. So many measurements and so many correlations. So really exciting work. So please go ahead and feel free to type your questions in the Q&A or raise your hand and we can call on you. I see we already have one question. It says few countries have different scales of AQI. How do you correlate the results from your device to the country-specific AQIs?
So right now with the AQI, we are only using the America Standards AQI.
I guess I'm wondering, you mentioned like putting this into an app.
Mhm.
What do you imagine the like user interface being like? How much information would a user need to or want to know, and how actionable would that be for them?
Yeah, so my thinking is that we can start with allergens. So if we see this street with our data, we see this street is full of pine pollen, then we will see this, we'll flag this area as a pine street, and people with pine allergies will try to avoid the street. So, but this will gradually roll out to more exposures. Also for some chemicals that can cause more instant response of human individuals. Some VOCs, for example. So we also have VOC data of a certain location to see, we need to avoid this street, which is harmful for everyone.
Sure. So we have a question. How are the different particles and biologicals detected separately by the device?
So we have a PES filter to capture those particles that we use for biological exposome analysis. So we extract the DNA and RNA from those filters and then use next generation sequencing to see what particles are in there. And for the chemicals, we have a zeolite absorbance to capture the chemicals, and those chemicals are sent to LC-MS to see what chemicals are in there.
And I guess beyond like recommendations of, okay, avoid this area because of maybe a pollen or allergen or whatever it may be. Are you thinking of also like creating kind of like a risk profile, given you have all this omics data and maybe, you're predisposed to this chemical causing this more than you than this other person? Or are you imagining incorporating that as well?
Yeah, that's excellent result. Yeah, we're thinking about it to create like a risk profile or giving a risk score of a certain exposures so that people can act upon those risk scores and to get better health.
Sure. It says many health outcomes may relate to chronic years exposure. Any plans for long-term monitoring?
Oh, yes. So this is a great question. So the studies we do right now are longitudinal profilings. So our participant will take one filter and then use the device for 2 weeks and then mail the filter back to us. So the entire study right now we have, the shortest one we have is 3 months. So we collect 3 months continuous exposures of those exposome components. We analyze their exposures and their potential impact on health. But of course, the longer monitoring will definitely help with the long-term chronic exposures. But this also comes with budgeting issues, like monitoring of people for like 2 to 3 years with this level of resolution is really costly.
I wonder with some of your studies, like, did you get any user feedback of like what form factor they would like it to be in? So like once you get the hardware electronics figured out, you could kind of make it into whatever form you wanted. Would they prefer like a watch, or did they like what you had, or what did you get a sense of like if they wanted to do longitudinal monitoring for a long period, they would have to want to keep using it. So I'm just curious what insights you've gained.
Yeah, definitely. Yeah, we'll talk to some of our participants, and so far the format's sort of okay, but a watch is definitely preferable. And some also mentioned they may want a clip so that they can clip on their belt. Yeah, so there are like lots of other forms they want to investigate. Yeah.
Great. Other questions? see. How long does the exposometer need to be exposed to the environment to detect a statistically significant signal? How quickly do the biological and chemical factors fluctuate in the exposome environment? Perhaps it depends on the factor being measured.
Yeah, that's a great question. So by nature, our samples are of low biomass. So that means even though we collected for 2 weeks, it's still the concentrations of those biological stuff are still pretty low. So the shortest we've done is to collect 2 days to get a reasonable readout compared with a background. But so with the device we have ourselves, the inter-device variation is quite small actually.
Another question is, did you think of adding radiation exposures like UV and ionizing radiations?
Yeah, that's a great question. Yeah, we do plan to have a UV sensor on board for next generation device. Also want to include a noise sensor so that we can now measure we can then measure 3 domains of the exposome, the biological and the chemical, and also with the UV sensor and noise sensor to measure the physical exposome.
Any other questions? All right. Well, thanks, Allison. Really exciting work. So many future directions you can go with this. So looking forward to seeing how this builds out in the future. So thanks so much for sharing all this work with us.
Yeah. Well, thank you for having me. I'm happy to project my research to other fellow researchers. Yeah. Thank you.
All right, everyone. Have a great day.